Welcome back to our second talk. In this session, we will have a short discussion about immunogenicity assessment by in silico and in vitro approach. Immunogenicity is the ability of biologics drugs to trigger patient's immune system. It will lead to generation of anti-drug antibodies that either neutralize the drug efficacy or result in faster drug clearance and shorter half-life. In certain cases, it may cause severe safety issues, including allergic reaction or CRS. It will put you in an impossible situation that you have to increase the dose because of the reduced efficacy and altered PK, but the adverse effect will not allow for escalating dose. Typically, ADA is generated in two different pathways. The T-cell dependent route is mediated by antigen-presenting cell, T helper cell activation, and production of anti-drug antibody by B cells. Alternatively, antibody complex or aggregates will cause receptor clustering, leading to generation of anti-drug antibody. There are multiple factors that could contribute to immunogenicity. In early R&D stage, product-related factors such as molecule sequence, structure, post-translational modification are the main players. During the process development stage, uh, formulations, impurities, and storage conditions are also part of the root cause. Later on in the clinical stage, patient-derived factors such as gender, age, pre-existing medical condition, and the therapeutic regimen, including dose, frequencies, and administration route, are the primary source of immunogenicity. As of nowadays, sequence engineering, such as humanization, is still the most well-established and widely accepted approach to reduce the risk of immunogenicity. With the climbing of percentage of a human sequence, people start off with murine antibody, followed by chimeric and the humanized version, end up with fully human ones. Generally speaking, fully human antibody does demonstrate a trending of relatively lower immunogenicity risk, as determined by the ADA incidence rate during the clinical trial. However, it does not offer any kind of a guarantee of free pass. Even for fully human antibody like adalimumab, the ADA incident rate is still as high as 49%. This data further supported the theory from our previous page. Not only the product, but also the patient and the treatment can cause immunogenicity. Immunogenicity assessment is a long-term effort throughout the whole cycle of a drug development. In discovery stage, people start with in silico screening to get rid of residues or motifs with liabilities, followed by a panel of in vitro assays to evaluate the potential immunogenic epitope and the T cell activation. In the meantime, both animal or artificial organ can provide more concrete evidence by in vivo or ex vivo models. During the preclinical stage, the focus is shifted from the sequence to product and process, including uh, uh, post-translational modifications, impurities, and formulations. Cytokine release assays and the non-human primate study are the gatekeeper that not only guide the optimization of a process, but also provides the baseline of a dosage for clinical trial. Finally, in clinical stage, patient and treatment-related factors will be evaluated. It typically includes ADA anti-drug antibodies, neutralizing antibody assays, patient HLA typing, cytokine release, T cell epitope, and T cell activation. In the following discussion today, we will focus on immunogenicity assessment by in silico and in vitro approach, starting with computer-based T cell and B cell epitope prediction. It is a prediction purely based on the sequence. You can do ranking of your candidates based on the number and the risk of the T cell epitope. Some people may also carry out deimmunization by mutations to remove these T cell epitope. However, these mutations within the CDR regions may have significant impacts on the antibody affinity and the function. The second checkpoint is the dendritic cell activation assays and cytokine release assay. It is a measurement of early sign of immunogenicity, but it is not equivalent to the actual immunogenic reaction. To take it to the next level, we need to evaluate the T cell activation by several PBMC-based assays. CD4 positive T cell PBMC assay is a relatively straightforward approach. However, it cannot differentiate the activation signal driven by either the T cell epitope immunogenicity or T cell agonist antibodies like CD3 antibodies. Peptide assay is widely used for the screening of a potential T cell epitope peptide. All you need to do is to incubate the peptide candidates with the PBMC, followed by measurements of T cell activation. DC and the T cell assays, the dendritic cell and the T cell assays, mimic the whole signaling pathway of the T cell dependent immunogenicity as described at the very beginning. We will share more detail about this approach during the case study sessions in later slide deck. Lastly, to pinpoint the exact sequence of T cell epitope, MAPS assay is the most suitable option to identify the processed and presented epitopes, but it heavily relies on the mass spec and analytical capabilities. There are several publicly available databases that provide DIY services for T-cell epitope predictions. These databases like IEDB will return a list of peptide sequences based on predefined HLA subtype and the length of epitope. Probal's team has established a comprehensive prediction package to combine the scores from different databases with percentile rank. For B-cell epitope prediction, the in silico analysis will return the potential linear and conformational epitope based on sequence or 3D structure. With the recent explosive development of AI technology, the immunogenicity assessment is taken to a whole new level. In addition to the database approach, as I mentioned in previous page, we are also using a software called WeAdapt from Wemo, an AI company that combines structural biology and deep learning models. In this system, besides epitope peptide binding affinity, a bunch of indicators including APC process, presentation probabilities, selection of HLA subtype, immune tolerance will be taken into considerations. This platform showed a decent correlation with clinical observation of ADA positive rate. Next, we will share more technical details about DC and T cell assays for in vitro immunogenicity assessment. The reliability and the quality of in vitro immunogenicity assessment heavily relies on the diversity of PBMC samples. Probal's PBMC bank comes with the capacity of more than 5,000 donors with similar distribution of a frequency as the real human population. To carry out the DC and the T cell assay, monocyte from PBMC is first isolated and differentiated into immature dendritic cell, followed by loading with test articles like antibodies or proteins. The mature dendritic cell is then incubated with CFSE labeled T cell isolated from the same batch of a PBMC. And the proliferation of CD4 positive T cell is analyzed by flow cytometries. It is crucial to use the monocytes and the T cell from the same donor and the same batch. Otherwise, it will trigger the immunogenic reaction, leading to a significant amount of background noise. As the T cell proliferates, you will see increased percentage in the Q1 quadrant, which is CSFE negative T cell, and decreasing percentage in the Q2 quadrant, which is CSFE positive T cell. And this is how we do the calculation of the proliferation rate for the CD4 positive T cells. To validate the assay system, we tested several reference samples, including two hyperimmunogenic antigens, PPD and KLH, and also two humanized monoclonal antibody, barcocizumab and pembrolizumab, as well as a fully human monoclonal antibodies, evolocumab. Both PPD and KOH, which gives 100% and 80% response rate in the PBMC, showed extremely high response intensities, while those three clinically validated therapeutic antibodies showed much lower response. Furthermore, the assay readout is also consistent with the in silico prediction by WeAdapt software, as I mentioned in previous page, and also the clinical ADA positive rate. In addition to antibody, we also tested this assay with endogenous proteins containing Fc tag against 3 donors, and only one is responding with low range of response intensities, as determined by the proliferation rate of the CD4 positive T cells. Finally, we use this assay to evaluate the success of humanization. Comparing to the parental VHH before humanization, 2 out of 3 humanized candidates demonstrated minimal risk by both DC and T cell assays, and also the in silico prediction, while the remaining VHH3 humanized version is considered as a failed design because comparing to the parental, both the in silico score and the percentage of immunogenicities is not changed a lot. Okay, that's all for the second talk. Let's move on to the Q&A session.